Welcome to Exam Snaps.
Name the bond connecting adjacent water molecules. Answer is hydrogen bond. Name the bond which connects hydrogen and oxygen in a water molecule. Answer is covalent bond. Explain the dipole nature of water.
Water has unequal distribution of charges. The oxygen atom is slightly negative and hydrogen atom are slightly positive.
What is meant by the term polar?
A molecule containing dipole, unequal sharing of electrons is called a polar molecule.
Complete the diagram to show two water molecules around a sodium ion when sodium chloride dissolves in water. Explain the importance of water.
Polar solvent. Water is a polar molecule, allowing it to dissolve many substances. This makes it an excellent solvent for ions and polar molecules. Transport medium. Water transports dissolved substances in organisms. Examples: blood in animals and xylem, phloem sap in plants. Medium for chemical reactions. Most metabolic reactions occur in aqueous solutions within cells. Cohesive and adhesive properties. Cohesion between water molecules and adhesion to other surfaces enable capillary action, essential for water movement in xylem vessels.
What is the general formula of monosaccharides?
Carbon from carbon dioxide, hydrogen from water and oxygen from carbon dioxide.
Describe the formation of a disaccharide taking maltose as an example.
Maltose is a disaccharide made up of two glucose monomers. The two glucose molecules join by a condensation reaction, forming a glycosidic bond and releasing one molecule of water.
Complete the equation. Glucose and fructose form sucrose and water. Explain why monosaccharide and disaccharide cannot be stored as a source of energy.
Monosaccharides are highly reactive. Monosaccharide and disaccharide are soluble in water, so that they affect the osmotic balance of the cell.
Explain the advantages of storing polysaccharides as storage form of energy.
Polysaccharides are insoluble. They WILLNOT affect the osmotic balance of the cell. They are physically and chemically inactive. They have a compact structure, so more glucose can be stored in a small space.
Name the organelle which stores starch.
Answer: amyloplast.
Name the monosaccharide composition of the following disaccharides. A. Lactose. B. Sucrose. C. Maltose.
Lactose is made up of glucose and galactose. Sucrose is made up of glucose and fructose. Maltose is made up of two glucose monomers.
Explain why amylase enzyme is able to break only 1-4 glycosidic bonds.
The active site of amylase enzyme is complementary to the 1-4 glycosidic bond only. 1-4 glycosidic bonds fit into the active site.
Compare and contrast the structure of amylose and amylopectin.
Similarities. They both contain alpha-glucose. They both contain 1-4 glycosidic linkages. They both contain carbon, hydrogen and oxygen. Differences. Amylose is unbranched whereas amylopectin is branched. Amylose doesn't have 1-6 glycosidic linkages whereas amylopectin has.
Compare and contrast amylopectin from glycogen.
Similarities. Both contain alpha-glucose. Both contain carbon, hydrogen and oxygen. Both provide energy. Differences. Glycogen contain more 1-6 glycosidic bonds than amylopectin. Glycogen is stored in liver whereas amylopectin is stored in amyloplast.
Name the bond connecting glycerol and fatty acids in a fat molecule.
Compare and contrast saturated and unsaturated fatty acids.
Similarities. Both contain carbon, hydrogen and oxygen. Both contain covalent bonds.
Differences. Unsaturated fatty acids contain CC double bonds whereas saturated fatty acids contain CC single bonds. Unsaturated fatty acids have fewer hydrogens. Unsaturated fatty acids are kinked, bent whereas saturated fatty acids are straight, linear.
Explain the reaction where two amino acids join together to form a dipeptide.
Condensation reaction where water is removed. Peptide bond forms between carboxylic and amino group. Dipeptide is formed.
Describe the secondary structure of a protein.
Secondary structure of a protein contain alpha helix and beta pleated sheets, held together by hydrogen bonds between the amino acids.
Describe the changes happen when an enzyme is heated above its optimum temperature.
Hydrogen bonds in the protein chain break easily. Enzyme denature. Active site and substrate are no longer complementary. No enzyme-substrate complex formation. No product formation.
Describe the characteristics of soluble, globular proteins.
Globular proteins contain both hydrophilic and hydrophobic side chains. The hydrophilic side chains face outward, interacting with water, while the hydrophobic side chains are folded inward. This arrangement allows globular proteins to form stable colloidal suspensions in an aqueous environment. They remain soluble and do not settle or separate easily.
Describe the characteristics of insoluble fibrous proteins.
Fibrous proteins consist of long chains of amino acids with a repeating sequence. They contain a high proportion of non-polar R groups, which are located on the outer surface, making the proteins insoluble in water. The parallel polypeptide chains are linked by hydrogen bonds, forming strong, stable fibers.
Describe the functions of glycoproteins.
Glycoproteins help proteins to hold water molecules, keeping tissues moist and flexible. They also protect proteins from digestion by proteolytic enzymes. In mucus and synovial fluid, glycoproteins act as lubricants, reducing friction between surfaces.